Okay, welcome to topic 7, which is nucleic acids. Nucleic acids are comprised of DNA and RNA. We're going to focus a ton on DNA today and in this video and the next video, but this whole unit has DNA structure, replication, transcription, translation, and we're going to touch on proteins and enzymes because you do need to know the ins and outs of proteins and enzymes. But to start, we're going to get into DNA structure, the scientists that are responsible for determining what DNA is and its structure. So let's get started. So with the basics of DNA, right? DNA stands for deoxyribonucleic acid. It is the recipe book for literally everything, functions, it's codes for all our genes. It is everything for a living organism. The credit of the discovery does go to Watson and Crick. Even though they got a lot of help from Franklin and Wilkins, the actual discovery does go to Watson and Crick. That is because they published their paper first, and they are recognized as the people responsible for it. But this discovery was an example of cooperation and competition because Watson, Crick, Franklin, and Wilkins did work together, though they did separately compete to publish their paper first. And internationalism. So especially between Watson and Crick, one is in the US and one was in England, so it was international, the relationships that these scientists had. Another group of scientists that are responsible for determining what DNA is Hershey and Chase. So they were responsible for actually determining that DNA is the genetic material in cells. So they did 2 experiments and they radio-labeled viruses. In one, they radio-labeled the protein code, and in the other, they radio-labeled the DNA. And then they took these viruses and injected them into bacteria. And then when they looked at the bacteria, they were able to determine, okay, when we just radio-labeled the proteins, nothing happened. But when we radio-labeled the DNA, our bacteria is now radioactive. They were also able to do this with looking at radioactive sulfur and phosphorus, and so determining what was on and in the proteins and the DNA itself. They centrifuged it down and then looked at where the radioactivity still was. And in the radioactive sulfur version, the radioactivity was in the supernatant, which makes sense. That's the liquid, which shows that it was not not where you wanted it to be, because the genetic material is in the pellet, which is at the bottom. And so when they did the radioactive phosphorus, which was coded to the DNA, they saw that the radioactivity was in fact in the pellet. The conclusion to these, of course, is that proteins are not genetic material and DNA is the genetic material. That makes sense, right? You know that DNA is genetic material. We talked about these 2 previously, but Franklin and Wilkins, they were the ones responsible for X-ray crystallography. They were the ones who took this picture on the lower left-hand side. This picture was used to determine the composition, orientation, and shape of DNA. And the way that they did this is they shot an X-ray beam through a crystallized DNA molecule, and then looking at kind of the wall that it was being beamed onto, they were able to determine the shape, because you can see like it would get, the X-ray lights would get stopped on the DNA itself, but be able to shoot through all these empty spaces. And so they were able to determine that DNA was double-stranded, had a backbone, a phosphate backbone, and twisted at regular intervals, giving it that helix shape. So let's talk about some key structure points of DNA. As I just said, it's a double helix, right? 2 strands that twist around each other. Each strand has nucleotides, has a sugar phosphate backbone, and the nitrogenous bases of the 2 strands are joined together via hydrogen bonds. There are 4 different bases, A, T, C, and G, and they are complementary to each other, right? A goes with T, C goes with G. The reason we know that is because another scientist, Chargaff. So Chargaff came up with these base pairing rules, and he was able to figure this out because of the size allowance in between the backbones. So he knew that you had to match up a purine with a pyrimidine because that was just the amount of space that they had. If you did purine and purine, it was not enough space, and pyrimidine and pyrimidine, it was too much space. So if you did 1 and 1, it was the correct amount of space in between the backbones of the DNA. So what is a purine and a pyrimidine? Well, a purine is like A and G. They have 2 carbon rings, so they're just a little bit larger, whereas a pyrimidine would be like T and C, 1 carbon ring, so just a little bit smaller. In general, a nucleotide is made up of the same basic things, right? You have a phosphate, a sugar, and then a base. Whether that base is a purine or a pyrimidine depends on what the base actually is, but in general, you have those 3 parts: phosphate, sugar, base. Again, you should know how many hydrogen bonds are connected in between these purines and pyrimidines. So you have 2 between A and T and 3 between C and G. So how are nucleotides connected? Well, nucleotides are connected from one to the other via covalent bonds, so a different type of bond. Whatever this sequence is that they line up in is the genetic code. And the genetic code is read in groups of 3, called codons. These triplets send instructions into the cell, and those instructions could be anything, right? They could switch genes on and off, make proteins, make enzymes, do anything. And so again, in between your nucleotides on the backbone are covalent bonds, and in between the bases from one base to another are hydrogen bonds. So we know that DNA is a double strand, right, of polynucleotides, many nucleotides. And we know that sugar phosphates, that backbone, is on the outside and the bases are on the inside. Those bases are held together by hydrogen bonds, that should say bonds. And the 2 strands run in opposite directions, called anti-parallel. And you can kind of see that in the picture, but we're going to talk more about that on the next slide. But you can see that like these pentagons face up, whereas these ones face down, so they are going in opposite directions. And that DNA is a double helix. So that double helix, the twist of it is held together by more hydrogen bonds. So let's talk about this 5' to 3'. Sort of touched on it, but let's get really deep into it. So again, reiterating that a purine must base pair with a pyrimidine. But they run, these strands of nucleotides run in a 5' to 3' direction. So 5', the reason that we call it 5' is because that is the end that's nearest the 5th carbon in the sugar. So the 5th carbon is like at the upper point of the pentagon. And so you can also see that it's connected to the phosphate. So this 5th point is the end of this like little piece of DNA that we're looking at. So this is the 5' end. Then covalent bond is the 3rd carbon is connected to the next 5th, or closest to the 5th. So you can see that the covalent bond goes from 3 to 5, in between 3 and 5, and then again 3 to 5, 3 to 5, and then at the bottom, the 3' end is where the 3 carbon is, but it does not have a connection to the next thing. So at the end, if the 3rd carbon is open, then that's the 3' end. And if the 5th carbon is open, then that's the 5' end. And then you have the opposite happening over here. 3' is at the top because it's got to be the opposite. 5' at the bottom because it's the opposite. So how is the double helix shape maintained? So we have hydrogen bonds holding multiple things together, right? It holds adjacent areas of the double helix. You can see that here. And it also holds together your nitrogenous bases. Complementary base pairs ensure that mistakes aren't made when copying DNA, right? If you have the same thing written forwards and backwards, you're less likely to make a mistake. The sugar backbone is hydrophilic, meaning that it's, it it likes water. It plays well with water, but the nitrogenous bases are super reactive, so they are protected on the inside of the DNA. So now let's get even more in depth. We're going to start talking about nucleosomes. So nucleosomes hold DNA together and form the frame of chromosomes. So here you have sort of just like a strand of DNA, and that DNA is going to wrap twice around a nucleosome, and then it's going to move on to the next nucleosome, and it makes a histone protein, and so you just like continuously wrap it, and then it makes a chromosome, which is just DNA held together by nucleosomes. But basically what this does is supercoil the DNA. And so you went from like a thin strand of DNA, and you just kind of wrap it, keep wrapping it, keep packaging it, keep coiling it until you can eventually form a chromosome, which is pretty small on its own, but it holds so much DNA. And so it's absolutely imperative that this DNA wraps around these histone proteins and wraps around these nucleosomes so that you can actually fit it into cells. So again, histone proteins allow the DNA to be supercoiled. It's 15,000 times more dense, so it's so compact. Think about how many cells you have in your body, and every single one of those cells has your DNA in it. Those cells, I mean, that DNA has to be so compact to fit in there. But when you are supercoiling DNA, these supercoiled sections can't be expressed because they're basically hidden away. It's like packing up your whole house and putting it in boxes, and then you just sort of, when you need to actually express certain genes, or in this case, use certain items from your house, you just unpack that one box instead of unpacking your whole house. So in this case, you supercoil everything, and then when you need to express genes, you uncoil that one specific area that you need to express those genes in, but you don't need to uncoil the entire piece of DNA. Okay, now we're getting to the tail end of this. We're going to start talking about types of genes. So there's single copy genes, which make up about 45 to 60% of the genome. 3% codes for structural genes, and the rest is spacer DNA. So there's two types of genes. There's exons and introns. These terms are going to be massively important, so you should absolutely write this down. Exons are coding regions, and you can think of it because exons exit the nucleus to go be coded by the ribosome. Okay? Introns are non-coding regions, so they can be edited, but they don't really leave. Then there is the other piece of DNA, which is highly repetitive sequences. They make up about 5 to 45% of the genome. So this is like sort of like the rest of it. This used to be called junk DNA because no one knew what it did, but science has improved, and we now have some idea of what this junk DNA does. Each of these sequences are repetitive, and they can be 5 to 300 base pairs long. And you can kind of see in the picture here, like, I don't know what red and green are, what they stand for here, but you could say that it's A and T, and it just goes AT, 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 right? All the way down. It's just repeating itself over and over again. Now, you knew I wouldn't leave you hanging on what junk DNA does, so previously it was called junk DNA, now it's called non-coding DNA, and it does do, there's a lot of things that it could do, but the kind of big thing that we look at are tandem repeats within satellite DNA and telomeres, which is repetitive DNA at the end of a chromosome. Telomeres are important because as you go through replication, sometimes there are mistakes at the end, and having all of these repeats at the end kind of protects against deterioration. So like you could lose a couple, say you had 300 A and T just repeating back and forth. You could lose like 5 of those and it would still be the same thing because you still have 295 of this repetitive DNA. So it's okay. So it does help against deterioration. Introns, like we said, non-coding sequencing. Non-coding RNA, so again, codes for RNA that are not translated into proteins, and then regulatory sequences, which are involved in transcription, and we'll get into that a little bit later, but the two big ones that I want you to focus on are satellite DNA and telomeres. And more so, I'm going to focus on tandem repeats. Oh my god, it's like I made this PowerPoint to flow so nicely. So tandem repeats are used in DNA profiling. So the way that they're used is within non-coding regions. People have really long stretches of repeating DNA called short tandem repeats. Short tandem repeats, that's what important to know. So when you take out those short tandem repeats, you can actually compare them between individuals because every single person that's listening to this video is going to have a different amount of these short tandem repeats in these different non-coding regions. And so that's what like DNA profiling and like anything like that, like when you're doing crime scene stuff, you compare these and you can determine, you know, who done it because individuals will have a different number of repeats within them. And that is it for today. You've made it through your first nucleic acid video. I'll see you next time.